Good day, everyone. I'm Lennon Ramos, and today I will discuss about the blood. First of all, the blood is a specialized fluid that is essential for life, and it is a liquid connective tissue which maintains homeostasis. The blood also transports nutrients, oxygen, and other waste products, which is the job of the RBCs. It also acts as defense against infections and diseases, which is made possible by the WBCs. The blood also clots to prevent blood loss, and we have the PLTs to thank for this. Overall, the blood is an important fluid in our body that keeps us alive and protected. So for the blood, there are four components that are in it. These are plasma, erythrocytes or RBCs, leukocytes or WBCs, and thrombocytes or PLTs. Let's discuss each one of them, starting with plasma. So what is plasma? Plasma is a straw-colored liquid that consists mostly of 91.5% water and 8.5% solutes. These solutes are mostly plasma proteins, which we will analyze later, and it also contains immunoglobulins, which are also known as antibodies. These antibodies attack viruses and bacteria and defend us from foreign cells. Now, let's analyze the plasma proteins. First is albumin. This protein is the smallest and most numerous plasma protein. It helps maintain osmotic pressure, which is important in the exchange of fluids across the blood capillaries. Next is the globulins. These are large proteins that produce the antibodies or immunoglobulins. And as I have said earlier, these antibodies attack viruses and bacteria. Last is the fibrinogen. This is a large protein that plays an important role in blood clot. This protein will be converted into fibrin, in which forms a mesh-like network of fibers that traps PLTs and RBCs to form a clot. We will discuss this further later in hemostasis. These three plasma proteins are mostly produced in the liver. Next, we have other solutes in the plasma. These are electrolytes, nutrients, gases, regulatory substances, and waste products. So what are these? Electrolytes help maintain osmotic pressure and is made out of inorganic salts. Nutrients are products of digestion, such as glucose and lipids, which are important for cellular functions, growth, and development. Gases are also important, such as oxygen, which is required for cellular activities, carbon dioxide, which is involved in the regulation of blood pH levels. Regulatory substances such as enzymes and hormones help maintain the body through catalyzed chemical reactions and metabolism, respectively. Then we have waste products like urea, ammonia, and bilirubin, which are transported by the blood for excretion. And that is it for plasma. The next cell we will learn is about erythrocytes or RBCs. In here, we will learn the anatomy, function, lifespan, and production of RBCs. First is the anatomy. RBCs have a biconcave disc-like shape, which is 7 to 8 micrometers in diameter. So basically, their shape is like a dome, but without a hole. Instead, the middle portion is sunken or it has a crater. RBCs are the most numerous blood cell, where we can see 4.8 to 5.4 million cells per microliter. To make it simple, imagine the population of Cavite during 2020, which is around 4.3 million. So to compare, Cavite is similar to a microliter. So just imagine that. There are more cells per microliter compared to the amount of people in Cavite. And to make it clear, a microliter makes about 2% of a drop of a small liquid. So in a single drop, there are at least 50 microliters. Imagine that a single drop of blood can have as much as 270 million RBCs in it. Another is that they lack a nucleus and other organelles, which means they cannot reproduce. That is why their body contains 280 million hemoglobin molecules, which are oxygen carriers and gives the RBCs its red color. Next up is their function. RBCs transport oxygen into the cells with the help of hemoglobin, and since they produce their ATP anaerobically, the oxygen they carry is not used, which maximizes the amount of oxygen they can deliver. Another function is that they regulate blood flow and blood pressure by being constantly on the move. While they circulate, they get 23% of the body's carbon dioxide, and brings it to the lungs for exhalation. Now for their lifespan, they only live up to 120 days since they cannot reproduce. That is why new RBCs are produced in the red bone marrows through erythropoiesis. Erythropoiesis is the process of making new RBCs, which is regulated by the hormone erythropoietin. Now that we're done with the RBCs, let's dive into the world of leukocytes or WBCs. Leukocytes are less numerous than RBCs. To put into numbers, for every 500 RBCs, there's only one WBC, since the ratio is 500, 30, and 1, where 500 is the RBCs, 30 is the PLTs, and 1 is the WBCs. So this just means for every 500 RBCs, there's only one WBC. So it's very small. About 1% of the blood's volume is WBC, but don't belittle this percentage, because that 1% is equal to about 50 to 100 billion WBCs in total. And since WBCs are the blood's enforcers, they are the ones responsible in defending the blood against pathogens, such as bacteria and viruses. Sadly, most of our WBCs live for only a few hours to a few days, because they are constantly involved in the body's immune response, and they undergo significant wear and tear during this process. And per microliter of blood, there are approximately 5,000 to 10,000 WBCs. Now these WBCs have five types. These are monocytes, neutrophils, eosinophils, basophils, and lymphocytes. I will discuss each one of them starting with the monocytes. So the monocytes are the largest type of WBC, which enlarges and differentiate into macrophages, which can be either fixed or wandering. And since they are phagocytes, they will commence phagocytosis, wherein they will engulf the pathogen and destroy it. But they take longer to respond and makes up of 3 to 8% of the total amount of WBCs. So for the parts of the monocyte, we have the cytoplasm, the nucleus, which is kidney-shaped or bean-shaped, and lysosomes, which degrades and recycles both extracellular and intracellular materials, which are the final steps in phagocytosis. Their diameter is around 12 to 20 micrometers. So that's basically WBCs are the largest among the five types. Now let's move on to the neutrophils. These are the police of the blood, since they are the most common WBC, which is around 60 to 70% of the total WBC count in our blood. They are also phagocytes and release lysozyme, which aids in phagocytosis. And they contain defensins, which direct antimicrobial activity, immune signaling activities, or both. For the parts of the neutrophil, we have the cytoplasm, multi-lobe nucleus, which is connected by chromatins, and granules, which release enzymes when your immune system is under attack. The size of the WBCs are 10 to 12 micrometers in diameter. Let's move on to the eosinophils. These WBCs enter tissue fluids to respond to allergic reactions, which is believed to release enzymes. And they also specialize in defending us from parasitic worms. The population of the eosinophils are around 2 to 4% of the total WBCs in our blood. So for the parts of the eosinophils, they are mostly similar to neutrophils. The only difference is that their nucleus are bilobed, which only means that there are only two lobes connected by a chromatin. Unlike the neutrophil, there are many lobes, 2 to 5. And their color is more orange rather than purplish. Their size is also the same with the neutrophils, which is 10 to 12 micrometers in diameter. Up next are the basophils, which is similar to mast cells, because they are both involved in the body's hypersensitivity and allergic reactions. These basophils are the least common type, which is around 0.5 to just 1% in the total WBC count of our blood. They release heparin, serotonin, and histamine in case our body has allergic reactions. So for their parts, we have the cytoplasm, nucleus, and granules. As you can see, they do not differ largely from the other cells. They are only darker in color and only 8 to 10 micrometers in diameter. The last type is the lymphocytes. These are the major soldiers in lymphatic system battles. But sadly, They only last a few hours in our blood, and since they last for a short duration, only small portions are present in our blood at a time, which is around 20 to 25% of total WBC population. So for these, it has three types. These are B cells, which recognize and bind to specific antigens marking them for destruction, T cells, which are responsible for cell-mediated immunity, and NK cells, or natural killer cells, which directly recognize and kill certain infected or cancerous cells without the need for prior activation or antibody production. For the parts of the lymphocyte, there are only two, the cytoplasm and the large round nucleus. There are two sizes of lymphocytes. These are small, which is 6 to 9 micrometers, and large, which are around 10 to 14 micrometers in diameter. Now that we have discussed these types, let's differentiate whether they are granulocytes or agranulocytes. But first, let's identify what these two are. Granulocytes are cells which contains granules that aid in our cell's defense mechanisms, and agranulocytes are cells that do not contain granules. So here they are, categorized and organized by size. Now I will show you a table to let you know what your complications may be if you have too high or too low levels of the WBCs. Take note that these are not exactly your complications. They only show what is possible. So, okay, let's move on to the thrombocytes or PLTs. PLTs breaks off from megakaryocytes, which becomes irregular dish-shaped cell fragments, which is around 2 to 4 micrometers in diameter, very small. They have many vesicles but no nucleus, and they are around 150,000 to 400,000 per microliter of blood. That is a lot. Now their granules promotes clotting, which then forms a platelet plug, which blocks off the circulation of blood in order to stop bleeding whenever we damage our body. These PLTs live up to only 5 to 9 days, and the old or dead ones are disposed by fixed macrophages in the liver, which are the Kupffer cells. Once our blood has been centrifuged, we will see layers in the tube. These are the plasma, Buffycoat, which consists of WBCs and PLTs, and at the bottom are the RBCs. Now let's discuss hemostasis. So what is hemostasis? In short, hemostasis is the process to stop bleeding. There are four stages when this occurs. The first one is vasoconstriction, which refers to the narrowing of blood vessels due to the contraction of smooth muscles in their walls. Next is the platelet plug, which I have discussed already. Then fibrin clot formation, which is a complex process that occurs in response to vascular injury or damage that adds an additional layer of seal to the platelet plug. Then last is fibrinolysis, where the breakdown of the fibrin clot is occurring once the wound has been healed. Now we go on to the blood types. Each individual has different blood types, so that is why we cannot simply get random blood from somebody and expect that nothing will happen to us. So there are four main types of blood. These are type A, type B, type AB, and O. And here's the chart of their antigens and antibodies. So the antigens are substances that are present on the surface of the red blood cells and can elicit an immune response from the body. Antibodies are proteins that are produced by the immune system in response to the presence of foreign blood antigens. Antibodies are specific to a particular antigen and can recognize and bind to it, leading to destruction of cells that display that antigen. So for example, your blood type is B and your blood has B antigens. So if you donate that blood to someone who has a type A blood, their antibodies will attack your blood, which will result in the destruction of your blood. So in short, your blood did not help that person. No. And to let you know, the type AB is the greediest among the four, since it has both antigens and no antibodies. This means that it can receive any blood, but it cannot give blood. The opposite of this is the type O, because it has no antigen, but has both A and B antibodies. This means that the O is a universal donor, but can only receive a blood from another type O. If you're wondering, how about the plus and minus in the blood, like A plus, wait, no, there's no A plus, A minus, B plus, AB plus? Well, these are caused by the RH of our blood. The RH factor, also known as the rhesus factor, is a type of blood antigen that's either present, which is RH positive, or absent, which is RH negative, on the surface of blood cells, of red blood cells. The RH factor is named after the rhesus monkey, in which it was first discovered. That is why you cannot also give an RH positive blood to an RH negative blood, which is similar to the functions of antigens and antibodies. Before we dive into the blood pathway, let us first briefly discuss about the heart's parts. These are the superior vena cava, pulmonary veins, pulmonary valves, right atrium, tricuspid valve, right ventricle, inferior vena cava, aorta, pulmonary artery, left atrium, aortic valve, mitral valve, and left ventricle. These parts of the heart will help our blood circulate throughout the body. This is also where oxygenated and deoxygenated blood passes by. So, let us first know what deoxygenated blood is. Oxygenated blood is rich in oxygen, which is a bright red color that delivers oxygen which delivers the oxygen that it carries to our cells. This blood became oxygenated when it arrived in our lungs. Next is deoxygenated blood. This blood is now low in oxygen and has a darker red color which delivers carbon dioxide to our lungs for exhalation and for the replenishment of oxygen. Now that we have discussed about the blood, let us know the blood's pathway. On the right, we will see Bogart, who will be our model for today. Let's take a closer look at Bogart. So for the blood pathway, it begins in our hearts. So in this case, Bogart's heart. So first is the deoxygenated blood entering from our superior and inferior vena cavas. Then it will reach the right atrium, highlighted in orange. Then the deoxygenated blood will pass through the tricuspid valve to reach the right ventricle. After that, it will enter the pulmonary valves, which will then pass through the pulmonary arteries from the left and right. Once it passes through those, it will reach the lungs, where the deoxygenated blood will be converted to oxygenated blood. Notice the change in color from orange to green, denoting oxygenation. Once the blood is oxygenated, it will return to the heart through the pulmonary veins, which will then go to the left atrium. Then, it will descend through the mitral valve to reach the left ventricle. Once it is there, it will pass through the aortic valve, then the aorta, then towards the parts of our bodies, from the top to the back, and even to the feet, to deliver the oxygen in the blood. Once the oxygen is delivered, the blood will then become deoxygenated, which will then return to our heart through the superior and inferior vena cavas. And then, the cycle repeats itself. And that is all for the blood cycle. Thank you, Bogart, for lending us your time. Now, for the last part of our discussion, we will learn about the six blood diseases. I will give at least two diseases per blood component. For the RBCs, we have thalassemia, which is a disorder where the body does not produce enough hemoglobin proteins, which is an important part of the RBCs. This will then result to the lack of oxygen supply, because hemoglobins are the carriers of oxygen in the RBCs. Take note that this disease can be inherited from your family. Another is polycythemia vera, where there is an increase in the amount of blood cells, but most especially RBCs, which will then make the blood thicker, which is similar to hemoconcentration. This will then cause a lot of complications, and if not treated, it will cause stroke and damage to tissues and cells. Now for the WBCs, we have leukemia, which is a cancer of the blood, but is more prevalent on WBCs. These cancers will cause abnormalities in our blood, which will then cause serious issues if not treated early. Take note that there is no cure for this, but it can be prevented and people do survive from it. This disease is common to old people around the age of 55 and above, and to children around the age of 15 and below. Another disease is lymphoma, another cancer, specifically in the lymphatic cells of our body. Remember that we have a WBC type called the lymphocyte. So this is a cancer of that WBC. And this cancer is one of the most common form of cancers. Notice that the disease are cancers because the dysfunction in the WBCs happen, which will then cause abnormalities. Now for the PLTs, we have thrombocytopenia, where the amount of PLTs in our blood is low, which will then cause problems whenever blood clotting happens, because there will be not enough PLTs to do it. This disease can last from days to years, but it does have treatment. Emphasis on treatment, because that does not mean it can be cured. And the last one is the Bernard-Soulier syndrome. This is caused by unusually giant platelet cells, thrombocytopenia, and prolonged bleeding time. This disease causes extensive bleeding, which can result to blood loss. There is no specific treatment for this, but it is not necessary at most times because care for patients is enough to help them overcome this disorder. And that is it. I hope you learned a lot from this topic. Once again, I'm Lenan Ramos. Thank you for watching.